that was just, the press release that just came out yesterday is sort of proof of that. So we can talk about that. So now I've given you a picture of embryonic stem cells, of nuclear reprogram, or sorry, of nuclear transfer. And the questions that scientists were really interested in that had to do with how does a stem cell, and when that stem cell does become a differentiated cell, what is happening to the genes in that cell or in the offspring of that cell? And is that something that we can manipulate experimentally? So this is where Yamanaka came in. So I just want to spend 2 more slides talking about sort of the mechanism behind this. So in this cartoon, this is just a somatic cell. So this could be your fibroblast from which the nucleus is removed. So this orange thing is the nucleus. And here in green is the egg that nucleus would be placed into. The idea is that in this somatic cell, the DNA here has proteins associated with it that are driving the genes that are specific to that lineage. So if this was a skin cell, then there would be proteins that are activating expression of skin cell specific genes. When you take that nucleus out of this context, so all these yellow blobs are the proteins that are regulating that, you take it out of that context and then put it into an egg context. All these blue things are the egg proteins that are specifying or regulating that embryonic stem cell identity and that genetic state. And so these blue molecules now can basically take over the nucleus that came from the differentiated cell and can convert it back to the embryonic stem cell fate. To think of that in a slightly different